So I'm very excited today to sit down with you and have a discussion with Dr. Brian Chait, the Camilla and Henry Dreyfus Professor at the Rockefeller University. Thank you so much for your time, Dr. Chait.
Sure.
I've been following your work for many years, back when I was a computational mass spec student at UC San Diego, and had the chance to e-meet you a few years ago when we did a panel together for science called Disrupting Life Science: How VHH Antibodies Will Shape Future Research, which provides a really great background on antibodies and VHH antibodies. So VHHs are an exciting modality of antibody. They're, of course, also called nanobodies. They're the variable region of the heavy chain only antibodies that are produced by camelids like llamas and alpacas. And over the years, we've noticed that much of the practical knowledge about VHHs is siloed and not necessarily accessible to new researchers in the field. So they have limited exposure to these molecules and their potential. And over the last few months, we've been collecting their burning questions. And so we would like to put some of them to you, Dr. Chait.
Sure, Natalie. Thanks. A pleasure to do it.
Yeah, thanks. So when we did the panel together, it was called Disrupting Life Science. And so I guess, there's this thought that VHHs are really transforming how we're doing science. And in your eyes, where do you see the biggest transformation happening, or where do you see VHHs having the biggest impact?
Right. So we originally got into it because we simply wanted inexpensive, high affinity GFP binding antibodies. And to use, because we do a lot of affinity isolation of, things like the nuclear pore complex, big objects, and we do biology on that and structural biology and so on. And so uh, the we came up with a method. I'll tell you a little bit about how that might have happened later. Um, and that worked pretty well. And we got scads of anti-GFP antibodies. And indeed, we were doing this as part of NIH funded research, and we made the plasmids available free to anybody who wants them, and hundreds of researchers have benefited from that. So we feel nice about that. That's a good thing. But it turned out it was pretty easy to do, and we came up with a method using mass spectrometry that evolved from some work that I was doing with some immunologists at Rockefeller. What actually happened, just for history, was that they were trying to find broadly neutralizing antibodies against HIV. And they were working with people who were infected with HIV but never got AIDS, never progressed to AIDS. These are called elite controllers, different names for them. Somehow their immune system just takes care of it. So that's a pretty interesting bunch of people. And for 20, 30 years, they'd been infected and never have any disease. And so my colleague, Michelle Nuzensweig, wanted to look at their antibodies and see whether there was something perhaps magical about it. And it's a long story, and he eventually succeeded beautifully and is in fact doing trials and things in Africa at the moment. And it looks like really quite a good contribution. But in the early work, they were struggling a little bit and were looking at memory B cells, which have these antibodies on the clones with these antibodies on the surface, and they couldn't get what they wanted. They were using single cell PCR to look at each individual cell, memory B cell, and that way you can get the heavy and the light chain and pair them nicely and so on. And anyway, they asked us to look in blood and see if we can correlate, what was being made by the plasma cells in marrow with these memory B cells. And apparently, even then, the correspondence wasn't properly understood. And so we looked in blood and used mass spectrometry and used as our kind of database what they had produced from memory B cells. And we found that there was quite a reasonable correspondence. And eventually, through a lot of very creative work on their side, they solved the problems and got their broadly neutralizing anti-HIV antibodies, which are now being clinically tested and can protect people for a long, long time and so on. But you know, while this was happening, I realized that you had to pair the heavy and the light chain, which is a hellish problem unless you're looking at a single cell. And so I thought, well, isn't there a way of getting around this? And I thought, oh, there's these single chain antibodies and there's no pairing problem. So we can do exactly what we did with them. And we would get lots of nanobodies, and maybe we can get high affinity nanobodies against almost anything.
Mm-hmm.
And that turned out about right.
Mm-hmm.
You know, so that was the basic idea. And you know, and I got good advice from Michelle on lots of things.
Mm-hmm.
He's a good immunologist. And he told me not to mess around with blood, but go and get some marrow, because
Okay.
lots of plasma cells in there. That's a very high density of them, and they're making they're making the antibodies we're using. Those are quite matured, and so on. So we did that, and we've never looked back.
So the bone marrow repertoire is something that not a lot of people look at in
Yeah,
llamas and alpacas. And I guess some way people get around that is by timing their bleeds so that maybe they're capturing plasma blasts as they rise after a boost.
yeah.
But I guess how do you see the serum correlating more with the plasma cells than with the memory B cells or with the plasma blasts?
To be honest with you, except for that early work, and that was, you know, that was around 2010, we haven't really looked at the correlation. It just works so ridiculously
Yeah.
well, so easily. And we work with a very nice company who looks after the llamas, and they're
Hmm.
professional. And they have vets that um, can draw bone marrow very nicely, and you know, and not cause too much discomfort for the animals. And you know, although they have to space these apart a little bit, that hasn't turned out to be much of a difficulty for us. So
Hmm.
we just get this incredibly good database almost so easily. And I remember when we began, we were struggling with blood and beta cells, and you know, and we weren't doing that well. I'm sure we'd do better now because we understand more. But
Hmm.
it works so ridiculously well. And as I say, Michelle says there's thousands of times more there than if you look in blood. So
Hmm.
don't mess around. And you know, I've always wanted to go back and do it, but we've never had the energy to do that.
Oh, okay.
Just works. It just
Yeah,
works
right.
so
If it's not
well.
broke, why fix it, right?
Exactly. And it looks like very unbroke. You know,
Yeah.
we get marvelous marvelous libraries.
Yeah. So then so you take an immunization based approach to discovery. And one thing that our community is often asking is why does it take so long to immunize a llama or alpaca versus a mouse where I could
Yeah.
do kind of a rapid immunization in maybe a month? Why do I have to take 10 plus weeks to get an immune response? I guess, have you thought at all about that?
Oh yeah, we've thought about it, but I got to say, you know, I'm not a biologist myself, um, and sort of humble in front of these rather complex questions. And the thing is that I think, you know, people shake their hands and say stuff, but I'm not sure, you can come up with rationales for it, but I don't think people know
Mm,
why the response tends to be a little slower.
yeah.
Or alternatively, why there are there's two, why I've often asked this question of zoologists who, know more than I do. My colleague Mike Rout, we've worked together on this stuff for a long time. He's a card-carrying zoologist and a good one at that. And I say, what's going on? And he said, camelids have been around for a long time, 15 million years or something, and they've all got it, and they've retained it. So it's clearly important for their lifestyle. They do better with this,
Mm-hmm.
and um, and uh, but exactly what it's giving them is still pretty much a mystery.
Yeah.
They live in, somewhat harsh environments, a lot of parasites around and a lot of different things they've got to protect themselves against. But we
Mhm.
sure don't have these little nanobodies, these
Yeah.
VHHs, yeah. Yeah.
Yeah. And then as you said, it kind of, the more you think about it, the more questions there are kind of because it is all one locus, the
Right,
conventional heavy chains intermixed with the heavy chain only antibodies, and it's basically different subsets of V genes and then different isotypes. What drives the isotype switch from G1, to G2 to G3 or G2B, G2C? The nomenclature here is also
right.
inconsistent sometimes. So, um, yeah, it keeps us up at night thinking about
Yeah.
whether or not how this is happening and how we can maybe do a better job of driving the immune response towards heavy chains. Um, I guess one thing I noticed from your papers is that you use both llamas and alpacas. And do you see a difference? Is there any reason to pick one versus another?
Yeah, I guess we've had relatively little experience of alpacas. I think we got uh, maybe 2 and 2 at one point. We've largely focused on llamas. And as I recall, um, the responses we were getting from the alpacas were a little bit weaker, maybe.
Mm,
But I don't think we had a good enough statistics,
mhm,
because animals vary one to the next. And, one trick that we came up with quite early on is all these animals get immunized against various things that could make them sick. And so when we look at what those responses are, and each animal is different. So the ones that respond well to that respond robustly to their immunization, we pick those ones for the animals that we will keep on and we'll take for our own and keep on doing experiments for many years. And they
mm,
live a long time,
mhm,
live as long as 20 years. And so I don't think I really have something deep to say about alpacas because our statistics isn't good enough,
mhm,
except that, we had a slight feeling that we weren't as good, but it may have been a luck thing. I got to say, the llamas are bigger
mhm,
and they're easier to bleed and get bone marrow from and so on. So, I think there's a tendency to that, but I think it was just that kind of early experience where we weren't overwhelmed with the response.
mhm.
But that could have been a blip,
Yeah.
so we just don't have statistics.
Yeah, we've definitely seen massive individual to individual variability to the point where some, might be producing 90% conventional antibodies and 10% heavy chain only, and those, of course, are to be avoided. And then some
Right.
that produce only, we encountered an animal that only produced the short chain, short hinge, and we
Yes.
saw zero of the long hinge. So that was
Yeah.
sort of, yeah,
Yeah,
there's,
we've seen things along those lines. There seems to be quite a bit of variability.
Mm-hmm.
Yeah, and that short and long is another mystery.
Yeah.
Because we don't know what that's about.
Mm-hmm.
We really don't.
Yeah.
We can
Yeah.
guess, but you can make hypotheses about all these things. But
Right.
to really test them robustly is quite hard.
Right.
Yeah.
Yeah. I guess, so you've immunized a variety of animals with a variety of different targets,
Yes.
from big globby things to kind of smaller things.
Yes.
Have you seen a difference in terms of targets? Do you think that are common factors that would drive more of a heavy chain versus a conventional antibody response?
Right. I think there's the obvious stuff like, when you get something that ought to be pretty immunogenic, like a viral component or so on. They often make crazily marvelous immunological responses. So, and when you get things that are very similar to the animal, you might get a somewhat lesser response. Although I'm always surprised and by how well it can differentiate self from non-self, even when there's quite modest differences. Yeah. But I would say the one thing that we've kind of noticed, again, the paucity of statistics makes me somewhat hesitant, but I think we're less good at making antibodies, VHH, against small things, really small things, like peptides. We've been pretty unsuccessful. We've wanted to make flag tags and all that sort of stuff, and we've really done not much good there. And we tried a bit, presenting it in different ways and so on. I wouldn't say we've been exhaustive by any means, but it was enough discouraging that we don't do it very much now. So that and there was a rationalization for that, and I'm not sure it's been tested properly. The conventional antibodies have a cleft between the light and heavy chain, and we said that's a good place for little things to, uh, be recognized. And you just don't have that cleft. The best you can do is, have a nice big CDR3 or something.
Mm-hmm.
But yeah, that's so we don't know, but we've not been very successful with little
Mm-hmm.
guys anyway.
Mm-hmm.
And so we have an inkling that they're not so good at that.
Yeah.
We sometimes imagine that and have a little bit of evidence that they certainly the nanobodies are good at getting in places. But in the actual animal, it's got an FC domain and it's still a dimer, so it's sort of bigger. And to what extent in that particular case they can get into little holes and what have you that antibodies can't get into is a good question.
Mm-hmm.
You know, certainly the nanobodies can. We've shown that many times that they can get into small places that have been trained, like the spike of SARS-CoV-2. We've looked at that endlessly, and through many strains, right? And that evolved in the whole world with billions of people, training it, and uh, and and uh, and still our little nanobodies that we originally made against the earlier strains are still many of them are still working.
Mm.
They can get in at conserved regions that just don't change in response to antibodies.
Mm-hmm.
That's kind of interesting. So
Yeah.
they're curious.
Yeah. Yeah. I guess one other concern that people often have is just affinity. They're worried that if they switch to a single chain antibody compared to a conventional, there might be epitope biases, but then there also might be a lower affinity and engagement. I guess, have you observed that, or do you think?
No, I think on the contrary, as long as we aren't looking at very small things, but decent protein domains, I would say that the affinities are terrific. And, one of the advantages of the mass spectrometric method, perhaps against compared to a display method, is that we pull these proteins out, these antibodies, the VHH out from blood with the antigen. And then we clean up the pull-out, the beads, we clean it up as stringently as we want. So we can treat it with madly stringent cleanups, like 4 molar magnesium or something. And we really try to clean it up. And only the very tightest binders can stay. And we find them. We find lots of them. And, as you say, you have to wait a while sometimes, and we keep on reboosting and coming back. And it just seems to get better and better. And the affinities that we get for proteins are every bit as good as the best antibodies. We get picomolar affinities, sub-nanomolar affinities all the time, lots of them. So we've been quite, on these trained animals, as long as you wait and so on, you can get beautiful affinities. And then if you want to go absolutely mad, you join a couple of them together and you get affinities that, they'd never let go.
Yeah.
These things are there for the duration.
Yeah. So one of the
They've got no off rate at all,
Oh, really? Yeah, just like
Well,
on your
not,
experiment.
they're so low that it's hard to measure.
Oh, interesting.
We originally did one of these for GFP, and we called it lobster claw because we got binding to all different parts of GFP, and then we took ones that were on opposite side and put a linker between them, and then that thing doesn't let go. It means you've got to have an off for both of them, and both of them are sub-nanomolar. And so you can imagine it just doesn't want to let go very easily. So those are very, very nice.
Okay. I was going to ask you because one of the benefits people talk about for VHHs is how modular they are, and you
Yeah.
can chain them together and make these bispecifics. I guess what was there an engineering challenge, or was it literally as easy as you say of, test pairs and pick ones that bind different epitopes and then they work?
That's what we do. And, we've not been very high throughput on that, but some of the people that we work with, who are trying to optimize the stuff for CAR T and stuff like that, are trying, large scale, combinatorial stuff. And yeah, you can find very nice things, but there's a lot of unknowns still. I mean, we still don't know, in each particular case, in which direction these things should be joined, how they should be joined, with what linkers. There's a lot of work to be done to optimize them. But the general thing we've found is that even when you take a monomer and you make a homodimer, you take the same nanobody and you just join it together, you invariably get better affinity. So you can start off with, pretty good affinity and then get very, good jumps. And if you put on, different epitopes, all kinds of interesting things happen. And so they're very modular and real easy to mess around with. And to join to whatever you want to join it to a toxin or something, or you want to join it to something that'll, just something interesting. You can do it, and the chemistry, often is not bad. It's not hard.
What's the most complicated molecule you've made with VHHs?
Oh, I don't know. I mean, we standardly make, let's say, a dimer or a trimer, and then we put another one on another antibody that nanobody that binds, let's say, albumin. And that then sticks around in a mouse for a longer time.
Mm-hmm.
And sometimes you want to tune the amount of time the thing is staying in there. Sometimes it's kind of good to have it. It's very toxic. You maybe want it not to stick around very long, to get cleared as fast as possible, bind where it should bind, and the rest go away. That's nice. But as often as not, you'd like it to stick there longer.
Mm-hmm.
And so you can tune the length of time by doing things like attaching it to bigger things or making polymers or attaching it to something like or putting an FC on it,
Mm-hmm.
which, recirculates and so on. So yeah, there's quite a lot and I wouldn't call us the best chemists on the planet. We just do really simple things that are easy to do, and you can do them. Making like I used to work with people who used to make biospecifics, and it's with normal antibodies, a bit of a job.
Mm-hmm.
And making them properly and getting knobs and things to work, all that, it's hard.
Mm-hmm.
And in this case, it's a piece of cake. You just put in the you dial up the LIGO and you're in business.
You're making it sound very easy, but it can't be that easy. So you've got VHH, linker VHH, tag for purification, maybe, and you're expressing it in E. coli, or what are you using to express it?
Yeah, we express it in E. coli, and we do it in. This is not my particular expertise because I never do it. Peter in our labs do it. We do it in a secretion system, and that seems to be good for folding and one thing and another. I don't know how much we've tested that, but it's one of those things that has worked quite well for us and gives us pretty decent levels that we just stayed with it.
Mm-hmm.
Some people use yeast and all kinds of things, but we've just stayed with this because it seems to be pretty robust.
Mm-hmm.
Our success level is high.
Mm-hmm.
So yeah, I mean, dialing up, buying oligos these days is inexpensive. And so we love that.
Mm-hmm.
We just, get a little bit of money for buying that and then pop them into a plasma and make it. Yeah. And
Yeah.
they work quite well. I'm sure that, I don't do any of that work. So the postdocs and so on do it. And occasionally I hear them having trouble doing something. And
Not often.
yeah, yeah, it may be something to do with the sequence if it's the same. I don't know,
Mm-hmm.
but generally speaking, it doesn't seem to be that hard. Seems to
Mm-hmm.
be that you have, and as I say, we're not the most creative sort of chemists that way, or recombinant protein makers in the world. So I have a suspicion that it's pretty okay. That's
Mm-hmm.
not that's not your biggest, you know,
Bottleneck.
yeah, bottleneck is the word.
Yeah.
Yeah.
So you've made a good argument for a mass spec based approach. So you're pulling out, higher affinity antibodies. You can be as stringent as possible with your purification. Why do you think not many people are doing a mass spec based approach? What do you think is the biggest roadblock for that?
It's like everything. It's you have to get a few chickens in a row. You have to have, a decent mass spectrometry. You have to have a little program to do things. So, you have all these things, and display methods in some ways are, you know, closer to molecular biologists. They do that sort of stuff. And so I don't think display methods necessarily are that much easier.
Mm-hmm. Doing it
Getting
well, for sure.
doing it well, getting big enough libraries into yeast, it turns out we do that as well, you see, and we have my comrades Fred Cross and Mike Rout are really good at this sort of thing. And they've really optimized it. I think they've done an awfully good job at optimizing it, making it simple.
Mm.
No, they don't go through all these steps of cell sorting or anything like that. They just go through one or two steps and it's done. And that's about as good as, and that way you keep a very big, uh, you keep a big variety of these things.
Mm-hmm.
Um, uh, when you choose too much, there seems to be, when you go to too many rounds, there seems to be uh, very nonlinear selections going on. And
Mm-hmm.
in the end, you just get a couple. And these folks do it nicely, I think. I think they took care and they have a very simple method that gets huge arrays of stuff. And I got to say, at least, we've mainly done that for very immunogenic stuff, like viruses. And we've done much less work on these things that are mildly immunogenic,
Mm.
which the mass spectrometry has worked awfully well on. So I can't say how it compares. But for the very immunogenic stuff, it's pretty good. And the big surprise for me was how well they did with affinity.
Mm.
Because in that particular case, on each, as I understand it, on each yeast, you've got literally thousands of nanobodies. So you can imagine that there's enormous effects,
Avidity. Yeah.
avidity effects, so
Mm-hmm.
that anything that binds a little should really come out nicely.
Mm-hmm.
But we still find a correlation between the numbers and that you get, how many times, and we count how many times you get that clone. And there's a nice correlation with affinity.
Mm.
And it works better than we have any right to believe it shouldn't work that. So it kind of works embarrassingly well, actually.
Yeah. That's good.
And the nice thing about that is you really get a lot.
Mm-hmm.
And so it's kind of fun when you get that much. You can do things on scale. For example, you can look at epitopes. You can put one set of nanobodies that you know that you've got that blocks. And so you can cut all those clones out, and you can do that on scale.
Mm-hmm.
And so we do that. We do that, and it works that works nicely.
Yeah. Kind of like a competition purification.
Yeah, yeah,
Pre-block. Yeah.
yeah, yeah,
Neat.
yeah,
So then I guess moving far away from molecular biology as possible, everyone's been talking about AI generated antibodies,
yeah,
and VHHs would be the lowest hanging fruit to do
yeah.
AI generation. Do you think we're there? Do you think we're almost there? Or do you think there's hurdles that the models aren't going to be able to learn easily that the llama immune system knows better?
Okay. Yeah. Well, I'm clearly not an expert on AI. There are some papers. I've read papers from David Baker and other people, and they always look, quite interesting. Um, what I would say from my own 2 cents on the thing, is that um, AI seems to work well when you got good training sets and you got big enough training sets. And, that's why I think it works so ridiculously well with AlphaFold and so on. So I think the training sets here are sparse at the moment. And so I think the key, if somebody wants to do well on this, is to do a bit of experimental work first and think of some clever ways of generating lots of data, or lots of high quality data. I think some creativity there is needed, and my guess is you can do it. But and there's different ways of getting different kinds of data. Uh,
Mm-hmm.
you know, but I think if one gets lots of good data and then uses that to help to train your AI, that would be helpful. And exactly how you get lots of very good data is a curious and interesting question. But I
Mm-hmm.
think there are some, there's some ways of doing that, and some of these
Mm-hmm.
ways don't give you necessarily fantastic, crystallographic. There's mutational methods that stop a thing from binding, and you can pinpoint where it binds. It's super easy, and you can do that at scale. Yeah. So I don't know how it will work, but no doubt this is very popular,
Yeah, it's
and
definitely coming.
and so it will progress. But I got to say, the animal's so good at it.
Yeah.
I mean, with 50 million years of evolution, and more of evolution, they're good. I, for the moment, I'm going to put my faith in them
Mm-hmm.
until you guys do better. You folks do better. Yeah.
Well, I think that is all my questions. I guess, was there anything else you wanted to add or bring up?
In thinking back on the last X years of nanobodies, um, it seems to me that the field has underachieved. I
Mm,
think that's the blunt truth. And because they're fantastic, having kind of worked with them, these little objects that you can engineer so beautifully and make so cheaply and are so robust. They're marvelous little, we try to destroy them, and it's so hard to destroy them. And so it would seem to me that they're just little things that attach to things. And they do the job fantastically, and they are easy to make and cheap to make. And so, why aren't they being used? We're using them quite extensively in research, in bio, pulling things out, and in crystal, in EM and all that, trying to lock things in particular states, or many things. But they really have, in my opinion, underachieved in as reagents for disease, for treating disease. And if you look at what antibodies are doing today, it's amazing. Half the drugs are antibodies. And so I think there's an enormous opportunity, which for reasons that I half know, but I don't fully know, uh, they've not done as well as they should. And
mm-hmm.
maybe things take just longer than we like, right?
Yeah.
Everything does seem to take a little longer.
I mean, I think when the caplacizumab was approved in 2019, I was of your mind thinking that, okay, now the path has been forged. Now we'll see lots of VHHs coming into the clinic, but you're right, it hasn't. And I don't know how much of it is just comfort with kind of the more proven path, or if people are trying to go after some of these popular targets with VHHs and not having the same success that they had with conventional.
Yeah,
But we'll see.
yeah. No, but there does seem to be lots of unfulfilled opportunities, for local things on skin, and for
Or yeah, delivery methods. Yeah.
delivery methods, and for things that are hard to get in, for, as I say, I think there's an obvious utility for CAR T and related techniques, because, all you're asking there is something that's got high affinity. And, what can do better than that?
Mm-hmm.
And so on. So I think there's lots of opportunity. And, the devil in this stuff is in the detail.
Mm-hmm.
And in sometimes in rational detail, and sometimes, as you say, in perhaps slightly irrational detail, things that comfort level, what have you. Yeah.
Mm-hmm.
But I think the future is bright.
Yes.
That's what I do think. Yeah.
Well, great. Well, thank you again so much for your time.
Sure.